Welcome to everyone from near and far, friends and family, and a special welcome to our examiners, Damon Clark, David Ginty, and Aravi Samuel. And it is my absolute pleasure to introduce Jasper. And it's a remarkable time for many reasons. It's unfortunate that we can't be in person, but I think one of the benefits is that we can have an array of people from near and far join us to hear about Jasper's work. And of course, today we get to hear about Jasper's work from him himself, about his efforts over the last several years. And he has done several projects, of which he'll only have time to kind of mainly tell you about two that center on motor control and motor circuits. For some background about Jasper, he grew up in Illinois and did his undergraduate work at WashU in St. Louis, graduating with his degree in neuroscience and applied math. And throughout those early years, he performed research on topics ranging from nanotechnology to membrane biophysics to cellular and neurobiology. And notably, after he graduated from WashU, Jasper worked with a nonprofit tutoring and mentoring underserved high school students in math and inspiring some students to consider moving on to scientific careers. And I thought this really nicely illustrated Jasper's commitment to things like social justice, which he continues today. When Jasper joined my lab, I was immediately struck by his thoughtfulness, ambition, and passion for science, although he also knows how to play hard and have fun. He's an independent and clear thinker, and as you'll no doubt experience, he's an engaging and skilled communicator as well. Jasper has what I would consider an insatiable curiosity and is on a constant quest for satisfying answers and meaningful understanding. And as such, he's kind of a perfectionist, and he will always optimize pretty much across everything. In the lab, he is what I would consider the super aligner, it sounds like things like aligning data is a trivial thing and maybe not so important and not something that you want to focus your attention on, but it's actually not trivial and hugely important for proper data analysis and just beautiful data sets, which you will see that he's created over his time in the lab. And as I mentioned, he's not all fun and games of science and data alignment. He also plays hard. He introduced me to several board games, including Codenames, which is fantastic. He's a Dance Dance Revolution master, I would say. A spike ball guru. He has years of playing soccer and rock climbing, and of course table tennis is his other passion and extreme talent. And he's the coach of the Harvard table tennis team. And when we're back in person, I will need to play you for probably the first and possibly the last time. But returning to science, Jasper has, I would say, pushed me as a mentor and as in terms of the field to the bounds of what we can do. Jasper was the first student in my lab, and I want to thank him for taking the risk of joining a new lab with a new investigator. I learned a tremendous amount from him about how to be a better mentor. We've had rigorous debates. I've learned how to communicate better, so on and so forth. And I've learned from him how I want to run my lab. And no doubt I still have a lot to learn, but I think the lab is off to a strong start in large part because of his efforts. So Jasper, thank you very much. And I turn it over to you.
Thanks a lot, Wei. That was a really nice introduction. Just checking, can everyone hear me? Wei, can you hear me?
Yes. Oops, sorry, hold on.
Okay, can you hear me?
Yes.
Great. All right, well, I'm really excited you all can make it to my talk today. It's really amazing to see so many faces here. I see not only members of the neuroscience community, but also some old friends, teachers, and professors. Many of you I haven't seen in quite a while, so yeah, really glad to see that you can make it. And I'm really excited to tell you about what I've been working on for the past couple of years. So I'm going to get us started by taking a look at some of the motor skills of some modern day robots. So robots can do pretty amazing things these days, whether that's the MIT Cheetah robot that's able to jump over barriers, or the Spot robot from Boston Dynamics that they recently taught how to open doors, or this hand robot from OpenAI that was recently shown that they could train it to manipulate a Rubik's Cube and solve it with only one hand. And so these tasks are all really hard, but they show that if someone really wants to design a robot to do one specific task, it probably can be done, and that's really impressive. But the number of complicated tasks like this that robots can do is still fairly short, especially compared to all of the things that animals can do. As some examples of this, whether that's a cheetah chasing down prey at high speed, or a monkey swinging through trees, needing to coordinate the movements of its arms and its tail to prevent from falling off, or a cockroach delivering a karate kick to the face of a predator to avoid being eaten, or a fruit fly that's able to cross a gap that's about twice the size of its body. All of these things are really impressive, and it's clear that robots still have a lot to learn from animals about performing these kinds of complex movement sequences. So to be fair, animals have had millions of years of evolution to fine-tune their bodies and their brains to be able to do these things. And so what I really want to do is kind of tap into these, the knowledge that nature has built through evolution to try to figure out how these systems work and tap into the expertise that animals have in their bodies and their brains. So I'm far from the first person to be interested in this. People have been studying motor control for over a century. So the nervous system is certainly what runs the show. Signals from the brain come down into the spinal cord. The spinal cord then organizes patterns of activity in motor neurons that project out to the body to make movements of the body happen. There are then sensory neurons that detect things like how much force is being placed on my foot, what angle is my knee at, and these sensory signals are then fed back, and they go both to the brain, but then they also go to these local circuits within the spinal cord called reflex arcs that are circuits of neurons that then use that sensory information to determine what the next proper movement is. And these can correct movements that were intended to go one way, but went another way. And so reflex arcs are known to be an important part of how the spinal cord works. This is a kind of closed loop control system where it relies on this feedback from the body to produce subsequent movements, but there's also a more open loop kind of control system that happens in the spinal cord called central pattern generators that can generate patterns of motor neuron activity without relying on sensory feedback. And so we know that these different parts are important because there are conditions in humans that damage them all, and all of them cause motor deficits to varying degrees. Whether that's Parkinson's or stroke that affects circuits up in the brain, or spinal cord injury, or ALS, also known as Lou Gehrig's disease, which causes specifically degeneration of motor neurons. Or even there are a couple of rare autoimmune cases where humans have had specifically their sensory neurons degenerate. And in all of these conditions, people lose their motor skills to varying degrees. And these conditions help us understand which parts of the nervous system are involved in motor control. And these conditions also make it clear why it's so important to try to understand the nervous system, because sometimes it breaks, and we'd really love to be able to fix it when it does. So just some examples of this. Stephen Hawking is one very high profile case of ALS, where his motor neurons degenerated and he was unable to move his body for his last many decades of life. Another example is Paul Selvin, another famous physicist who also happens to be my dad. About 15 years ago, he was in an accident where he was hit by a car while he was on a bike, and he ended up with damage to his motor cortex that caused weakness on the dominant side of his body. So one of my favorite things in life is playing table tennis with my dad. And he's had to learn how to play with his left hand, his non-dominant hand. And he's gotten really, really good with his left hand, and he can even beat me sometimes. But I think he and I both would be much happier if we could restore his ability to control the dominant side of his body. One of the really amazing or fascinating things about these motor disorders is, many of these have neurons damaged, but the body itself, the muscles, are mostly fine. Certainly they degenerate a little bit from lack of use, but for the most part, the motor neurons are, the muscles are fine. It's just the parts of the nervous system that are damaged. And so that makes us wonder, can we replace these kind of damaged wires? Can we build some kind of a replacement part and put it in to circumvent the damaged portion? And there are efforts to do this kind of thing, and probably someday it will be possible. But at this point, we don't really know enough about how we might design those kinds of replacement parts for broken parts of the nervous system, or how we might build a replacement part and then kind of connect it to the healthy parts of the nervous system in a way that restores function to human patients. And so we want to understand more about the details of what's going on in the nervous system. And so to understand how the nervous system works, it helps to know what the parts are inside of it and how those parts are connected. And this is, not a particularly novel kind of approach. If I gave you a computer or a car and asked you to understand how it worked, you would probably do something similar, where you would kind of look inside, see what the different parts are, and see how the parts are connected. And now this is kind of just, one level of analysis. There's many other ways that you could try to figure out how a nervous system works, but this is really one of the important tools that we have. So you might think, well, don't we know that already? Don't we know what the nervous system is made of? It's made up of neurons and glia, right? And it's certainly true that from hundreds of years of study, going back to Ramon y Cajal around 1900, we know what the shapes of many different types of neurons are. So on the left here is a pyramidal neuron in human cortex, and on the right is a Purkinje cell in the cerebellum. These are kind of two of the most well-known cell types in the mammalian brain. But while we do have a handle on some of these cell types, there are really just millions and millions of different types of neurons. And it's been difficult to tell what all of those different types look like, because they're so densely packed together in the brain that typically, in order to see the shape of a single neuron, you have to rely on sparse labeling to just, for instance, fill a single cell with a dye. So you can see that one cell, but that means you miss out all the rest. Neurons are also wired together into these really complex networks that make up your entire nervous system. This diagram is just showing about a dozen different neurons, with pyramidal neurons in cortex projecting down into the spinal cord and then stimulating motor neurons. But in reality, nervous systems have millions or billions of neurons, and understanding how all of those different neurons are organized into these complex networks has been a challenge. So while we have these kinds of partial maps of the different types of neurons in the brain, there are only a couple of tiny animals for which we really have a complete map of the nervous system. And by a complete map of the nervous system, I mean, we know what every single one of their neurons looks like, and we know how they're all connected to one another. And these kinds of complete maps of the nervous systems have been incredibly useful, and creating more complete maps of larger animals is a major thrust of modern neuroscience. So some of the animals that are kind of on the roadmap are the fruit fly with about 100,000 neurons, the mouse with about 100 million, and maybe someday we'll be able to get a complete map of a human brain with about 100 billion neurons. So with the way that progress is going, it looks like the adult fruit fly's nervous system is probably going to be mapped in the next couple of years. The mouse's nervous system may be something like 10, 20, 30 years away. And whether or not we can map a human's nervous system within the next century, maybe, but the technology that would need to be developed to do that, we don't really have a sense of what that would be yet. But there's a lot of efforts in the field to develop new imaging technologies to map larger and larger nervous systems. And so I've been really excited to be part of some of those efforts. You might notice that the animals in the top row, oh, my timer didn't start, that's okay. The animals in the top row don't have limbs, whereas the animals in the bottom row do. And so if we want to understand something about how the nervous system controls limbs, we need to map a nervous system of an animal that has limbs. And for that reason, the adult fruit fly is looking like it's going to be the first limbed animal with a complete map of its nervous system. And I've been lucky enough to be part of those efforts of mapping its nervous system over the past couple of years. So fruit flies are small, but they are mighty. So fruit flies, if you look at the back of a penny, fruit flies are about as big as the last 3 letters in the United States of America. So these are not the kind of bigger house flies that sometimes you see darting around your house when they get in. These are instead the tiny, almost microscopic flies that sometimes will hover around over ripe bananas if you leave them out too long. So despite the fly's small size, it's incredibly agile. I showed you earlier that they're able to crawl over these gaps that are twice the size of their body. They also are able to fighting over food, fighting over mates, and this requires coordination of the whole body movements, coordination of the limbs. And they also are incredibly fast. They walk at something like 10 to 20 steps per second, and coordinating 6 different legs, segmented legs, at step cycles of 10 to 20 steps per second is no easy task. And so all of these things really require a lot of coordination. And the truth is, we would love to have robots with motor skills as good as these tiny fruit flies. In fact, recently someone built a Drosophila-inspired robot called Drosophibot that mimics many of the aspects of the fly's body in terms of what its limbs look like and that sort of thing. But it can't yet display all of the kinds of complex behaviors that real fruit flies can, in large part because we don't know how to give it control systems that will enable it to perform these kinds of complex behaviors that we see real fruit flies do. And so to make some progress on this, we really want to understand more about how motor control happens in the fruit fly's nervous system. So fruit flies, they have a brain up in their head, just like us, and then they have a ventral nerve cord in their thorax. This is their equivalent of the spinal cord. The ventral nerve cord looks something like this. It only has about 20,000 neurons, and each of these spherical regions is predominantly in charge of controlling one of the 6 legs. And so the organization is kind of nice and simple. If you want to look at the neurons that control one of the 6 legs, you know generally where to look. You just look in one of these spherical regions. Motor neurons project out of the ventral nerve cord and into the leg. The legs have about 14 muscles in them, and there are only about 65 motor neurons that control each of the 6 legs. And compare that to humans who have thousands to tens of thousands of motor neurons per limb. And the fact that flies can do so many different athletic, mobile behaviors with this few neurons is really amazing. We don't know how the VNC does it, but because it's a relatively small circuit, only 20,000 neurons total, only 65 motor neurons per leg, we think we have a shot at really understanding in kind of a complete way what this system is doing. It turns out that the brain is maybe less necessary for controlling motor skills than you might think. So these are 2 videos of flies that have had their heads removed. So these flies don't have their brains, but they're still able to coordinate movements of their body. So this is what happens if you give a headless fly caffeine. It kind of freaks out and is starting to spaz out, but the spazzing out is not totally random. You know, this is not a seizure. This is coordinated movements of the limbs, taking steps forward, grooming its wings. And so it's clear that the brain is not necessary for generating some of these kinds of motor patterns. If you take a headless fly and tickle its leg, it will respond by doing this kind of grooming sequence to try to get the scientist to go away. And this looks very similar like it would look if the fly had its brain. And so not only can the ventral nerve cord generate motor output, it also can respond to sensory input and initiate actions. And we think this is just super cool. You may have heard the phrase running around like a chicken with its head chopped off. And that's because actually lots of animals don't need their brains to produce coordinated movements of the body. Obviously, the brain is important for many, many things. And these flies really definitely have a kind of reduced behavioral repertoire. But the amazing thing is that the ventral nerve cord actually takes care of a lot of this kind of motor coordination, this kind of motor intelligence that we'd really like to get robots to have. And so if we want to understand this, we need to look at the VNC. So we know that the fly's VNC is probably using these kinds of central pattern generators and reflex arcs, these open loop and closed loop systems. But exactly how those systems work for controlling limb movements is not really well understood. And because the fruit fly is so small, this is going to be a good system for us to go and try to answer questions about how these kinds of systems work and how they interact with one another. That's been a major challenge to understand interactions between open loop and closed loop control systems for motor control. So some specific questions, how are complex movements produced? How does the ventral nerve cord coordinate movements for the same limb? And how does it coordinate motor neurons that control different limbs? Another specific question we're interested in is which types of motor neurons are most influenced by sensory feedback? This is trying to get a sense that actually where are the reflex arc pathways? How do they interact with central pattern generators? So As I mentioned, the goal of my research project has been to completely map the adult fruit fly's motor system. And one motivation for focusing on the ventral nerve cord and the legs is that researchers at Junalia Research Campus are already making great progress on completely mapping the adult fruit fly's brain. Those efforts have helped in understanding the brain's sensory systems and navigational systems and memory systems. And I want to bring that kind of knowledge to the fruit fly's motor system. Some specific things that I worked on: I helped develop new imaging techniques for mapping nervous systems. I was then able to use those to map about 1,000 of the neurons in the VNC and their connections. And from that, we found a new reflex arc that connects the largest sensory and motor neurons. We're currently on our way to mapping all 20,000 neurons in the VNC to get a really complete map. And then as a second project, I worked on mapping what's actually going on in the fly's leg. You know, animals have had these millions of years of evolution to optimize not only their nervous systems, but also their bodies. And we want to see what kinds of cool things are going on in the fly's leg that might have been missed with previous mapping efforts that couldn't get a kind of comprehensive picture of what's going on. Okay, so with that, I'm going to dive into project one. So electron microscopy is a technique that allows you to zoom in and take pictures of a slice of the nervous system that reveals the nanoscale details. Here you can see membranes that outline the branches of different neurons. So these are cross sections through different neurons, axons, and dendrites. You can also see these bigger black blobs or mitochondria. And importantly, you can see synaptic vesicles and these structures that indicate the sites of synapses, or connections between neurons. So this larger neuron here is releasing synaptic vesicles onto these much smaller twigs of different neurons. And this indicates a site of connection between two neurons. So because it's so zoomed in, EM typically shows you these very small regions that makes it kind of hard to tell, well, so what is the shape of this neuron on a larger scale? And how many connections are there between these two neurons on a more complete scale? But new technology is allowing for much larger volumes to be imaged with electron microscopy. How we do this in our lab, which is similar to approaches taken in other labs, but has a couple of new developments on it that allow it to be faster, is we take a piece of tissue, we slice it horizontally into these ultra thin slices. We collect each of those slices onto this tape. We can then put that reel of tape into an electron microscope and project images, electron microscopy images of what each of the different pieces of tissue look like, and then align the many different millions of images that we take into a three-dimensional volume to end up with these complete electron microscopy volumes of a neural circuit that allows you then to go in and see all of the synapses and the shapes of all of the different neurons. So that's what it looks like in cartoons. What it looks like in practice is you dissect out a fruit fly's nervous system, you fix it with aldehydes, you then stain it with heavy metals like osmium, lead, and uranium, embed it in a block of epoxy resin so that it's hard, and then you can slice it very thin. You then slice off these very, very thin pieces of the tissue and collect them automatically in kind of a conveyor belt style onto this tape. You'll notice that this tape has these holes in it, and our goal is to collect each slice of tissue onto one of these different holes to enable us to take pictures of it. So at the end of the day, you end up with this rolled up reel of tape that contains many, many thousands of slices of a piece of tissue. And these slices are incredibly thin. To cut through one VNC, it took 28 hours of continuous automated cutting and collecting tissue sections. And yeah, it took 4,400 slices, each of which about 45 nanometers thick, which is about the size of a synaptic vesicle and 1,000th the width of a human hair. So once we have all of these slices collected onto a reel, we can put it into a custom electron microscope built by an engineer in the lab, Brett Graham. And we project an electron beam through the tissue slice and take pictures that look like this. And again, each of these individual images is of a fairly small field of view, very zoomed in so you can see synapses. But this microscope is able to then go and take many, many pictures of neighboring regions in a given slice of tissue to build up this big panorama of what a whole slice of tissue looks like at synaptic resolution, but with enough field of view to really see everything that you want to see. And the microscope can then go and do this for the next slice and the next slice and the next slice. And so for the data set that I was able to generate, this microscope captured 20 million images over about 2 months of imaging time. So this amazing microscope gives us the ability to get this high resolution images over really large areas. And I'll show you this video so you can appreciate the difference in scales needed to see synapses, the field of view of this smaller yellow rectangle, compared to the size of the VNC. And only once we have these really large panoramas or montages of a single section can you tell that, all right, this is now a full slice through the ventral nerve cord where we can see everything that's happening in this slice. And I put large areas in quotes because still this entire region is only about half the size of the letter I on the back of a penny. So synapses are incredibly small. Ventral nerve cord is very small in kind of our scale of things, but it's still quite large on the scale of electron microscopy. And so the ability to generate these kinds of large electron microscopy data sets has really, there's been a revolution in this field called connectomics in the past 15-ish years, where there are now these kinds of technologies emerging that allow you to map nervous systems that are reasonably large sizes. So after taking those kinds of images for all 4,400 tissue slices in the VNC, they can be aligned into a 3D volume. So here I'm showing you slice number 2000. And when I press play, we're going to step from slice to slice in this aligned image volume. And you can see each of the branches of the different neurons winding through the data set such that you can easily by eye follow where each of those branches go. This will allow us to then map out the shapes of all of the different neurons and map out all of the synaptic connections between them. But it still takes a lot of work to actually go and follow each individual neuron to map out its shape. And so an example of how we do this is, for instance, in my project, we started our reconstructions at a peripheral nerve. So we went to the region in the data set right where all of the axons were bundled together, about to leave the VNC and go out into the leg. And what this looks like is you get this slice through just hundreds and hundreds of different sensory neurons, bringing information into the VNC, as well as motor neurons bringing information out from the VNC to the legs. And so you just see a lot of these different circles, which are cross sections through different axons. And in order to reconstruct the actual shape of this neuron, we just put a dot in the middle of one of these tubes, and then go to the next slice of the data set, put another dot in the center of that tube, next slice, another dot, and so on, until we build up these reconstructions of the branching pattern of single neurons. So here I'm showing you the branching pattern of one of these sensory neurons that we reconstructed. This dashed arrow indicates where the sensory neuron's axon comes into the VNC. Out farther that way is the leg, which is not part of this electron microscopy data set. This light gray outline is the outline of the VNC, and this darker spherical region indicates that spherical part of the VNC that primarily controls one of the legs. So depending on which neuron you start at, you might get something that looks like this, which is a sensory neuron with a fairly simple branching pattern. Or it turns out if you start over in this region, you're going to end up with a motor neuron that has this more complicated branching pattern. Here, the sphere indicates the cell body of the motor neuron, and the axon of this motor neuron comes out of the cell body and then goes out towards the leg, while many different dendritic branches come off the side. And so we're able to do this for then all of the neurons in a peripheral nerve and build up this complete catalog of cell types that connect the VNC to the leg. And I want to emphasize that this is the first time that complete mapping of neurons controlling a limb has been possible. So we saw that these neurons come in a few different types of branching patterns, and here neurons with similar branching patterns are shown in the same colors. And I'll get more into the different types in just a minute. So that's what happens if we do reconstructions of everything in the peripheral nerve that's associated with one leg. But we can then zoom out and do the same kind of effort across many different peripheral nerves in the VNC to build a more complete map of, for instance, all the different types of motor neurons. So we did this for a bunch of different peripheral nerves and managed to find all of the different motor neurons that control the different limbs as well as the neck. And it turns out there aren't actually a ton of motor neurons. There are only 507 across all of these different segments. And so we were able to actually go and at least partially map out every one of them to get this kind of complete count. And it turns out the front legs are controlled by about 70 motor neurons each, whereas the middle and hind legs are controlled by closer to 60. In contrast to this relatively approachable number of motor neurons that we were able to map out, there are a ton of sensory neurons that enter the VNC, something like 7,000. And so at this point, we've only had time to map out about 650 of them. So we're about 10% of the way there. But we have the data set to be able to go and map out the rest. So after getting these reconstructions, we then registered them to a standard atlas of the flying nervous system. And this enables us to compare the reconstructions that we did with previously published images of the VNC's neurons, because many people have been doing mapping of neurons in the VNC before. It's just they've always been these partial maps, looking at a couple cells at a time, never really been able to get this kind of complete picture. And so here's some examples of some previously published neurons. So a previous study showed that sensory neurons with a morphology that look like this, these are sensory neurons that encode vibration of the leg. Sensory neurons with a morphology that look like this, they encode the angle of the joints. For instance, what angle is my elbow at? And sensory neurons that have a branching pattern that looks like this, they encode the velocity of the joint, movements of the joints. And so because previous studies have found this, we were then able to go and look in our EM data set for neurons with similar shapes. And we use an algorithm called NBLAST that helps assess the similarity between the morphology of different neurons to help us with this process. And indeed, we were able to find many neurons in our EM data set reconstructions that match the morphology of these previously published neurons. And this is exciting because that means that now we know that in the EM data set, these neurons encode vibration or joint angle or joint movement. And now we can actually go and map out their synaptic connections to figure out how they send information to other types of neurons in the VNC. So in addition to finding known types of neurons like these in the EM dataset, we also found some new neurons. So this was a type of sensory neuron that is the only type of sensory neuron that projects its axon bilaterally. So again, these dashed arrows indicate where the sensory neuron comes in into the VNC. And so you'll see that there are sensory neurons like this that come in from each of the 3 different legs, and then they have this really wide branching pattern that goes to different regions of the VNC. And so yeah, these are the only sensory neurons with bilateral projections. And also, they're the largest diameter sensory neuron. So their axons are really huge relative to the size of other sensory neurons. And we found that there are exactly 2 neurons with this kind of morphology that come in from each of the 6 legs, which means that in total, there are 12 of these neurons. And these conclusions that I'm telling you about were only possible because we were able to do this complete mapping effort. And that let us see all of the sensory neurons and make conclusions about, this is the only sensory neuron with a certain property. It's the largest. There are exactly 12 of them. And so these kinds of complete mapping studies give you a kind of information that you couldn't get with only partial mapping efforts. So although the morphology of these neurons was fairly new, by looking at similarities to other types of sensory neurons, we're pretty sure that this is a type of sensory neuron that detects how much weight is placed on each leg. So, do I have more weight on my left leg or my right leg? We think that these sensory neurons detect that kind of information. We also noticed that in something that's fairly unusual for sensory neurons, they had branches of their axons that run right alongside motor neurons. So here in gray, I'm showing the main branches of motor neurons with cell bodies that then project their axons out to the periphery. And here in this box region, you see that there are branches of these BCS neurons that run directly alongside them. And this is true both for BCS neurons that come in from the front left leg, as well as neurons that come in from the other leg. They also send a branch to this region to run directly alongside motor neurons. And so we asked, well, maybe there's a direct synaptic connection here. And because it's an electron microscopy data set, we could just look and see whether there were direct connections between these BCS neurons and motor neurons. And indeed, there are tons of direct connections between the BCS neurons and the motor neurons. In fact, the motor neurons are the main synaptic output of the BCS neurons within this region. And 98.6% of the synapses were made onto motor neurons. So we then looked at other regions of the data set to see if this trend was true for other legs, and indeed it was. We didn't do it as complete of a mapping, but we did see that all of these BCS neurons make direct synapses onto motor neurons in each of the different regions of the VNC to which they project. And so what does this mean? This means that this is some type of sensory neuron that seems to be able to coordinate simultaneous movements of multiple legs by directly stimulating motor neurons controlling multiple legs. The other thing is that these synapses are placed onto the motor neurons right around where we think the spike initiation zone of the motor neurons is. So we think that they're really poised to drive activity in these motor neurons to really quickly bring about movements. So the next thing we want to know is what types of motor neurons receive these synapses. It's not the case that every motor neuron receives synapses from the BCS neurons. Turns out that there were only a couple of different neurons that really received strong input from them. And so we wanted to know, what were those neurons? Which muscles did they control? What kind of movements did they make? And so to get a handle on that, first, let's just look at what the motor neuron reconstructions look like. So again, here we have motor neurons with cell bodies up here, an axon that then projects out to the leg, and dendritic branches that come off. And what we noticed was that motor neurons are really symmetric. This is something that had been seen certainly to some degree before, But notice here that in this pair of motor neurons, they have a really similar branching pattern, a very similar divergence here, a couple branches coming up, a couple branches coming down. And this is not the only example of motor neurons that come in these really symmetric pairs. It turns out almost every motor neuron has a morphology that has not an exact match, a rough match on the contralateral side, and no other motor neuron on its own side tends to look like it. So again, these motor neurons are not perfect mirror images, but from our analysis, we thought we were able to see that about 80% of the motor neurons have branching patterns that are identifiable enough to recognize them on the contralateral side of the body. And so we hoped that means that these branching patterns would be unique enough that we could then compare them with images of previously published motor neurons in the same way that we did for the sensory neurons. So we went and looked at images of previously published motor neurons with known properties. For instance, here's a motor neuron that controls, that generates really high force movements of the fly's equivalent of a wrist. This is a motor neuron that controls the same joint, but generates only small movements when it's active. And then this is a different motor neuron that controls the equivalent of the fly's fingers. And so we just looked, can we find neurons in the EM data set that have morphologies that are really identifiably the same as these previously published neurons? And again, we use NBLAST to help us out with this to generate similarity scores between our EM reconstructions and the previously published neurons. And we were able to find pretty good matches in a lot of cases. And astonishingly, it turns out that some of the really high force motor neurons receive lots of synapses from the BCS neurons, whereas other types really don't receive any. And so here's a video showing that. So again, the BCS neurons coming in from the left side and the right side, making synapses onto one specific high force wrist flexor motor neuron, whereas not making any synapses onto this yellow neuron. So this has been really exciting to map out, but what does this reflex arc actually do? You know, what actually happens when this pathway is stimulated? We don't have an answer to that yet, but thanks to work of Sweta and Anne and John Tuthill's lab, we now have genetic driver lines that provide access to this cell type so that we can, in the future, do things like activate or inactivate this type of neuron and see what behaviors the fly does. So that's the first set of results to come from this EM data set that I generated, but hopefully it won't be the last. This EM image data set is now publicly available, and we're helping coordinate the efforts of something like 10 different labs to reconstruct different neurons and their connections within this data set. Here I'm showing you the progress of this reconstruction community as of September of last year. And these images don't include any of the 1,000 neurons that I mapped in my project. So really there's been a lot of other mapping efforts that have gone on in this data set. So everything I've told you about so far has involved really painstaking manual reconstruction of the branching patterns of each neuron, and we want to speed this up. And thanks to recent advances in machine learning and computer vision, and we're working with a company called ZAI to perform this, we can automatically reconstruct the morphologies of different neurons. And the way that this works is instead of just putting kind of a line down the middle of each neuron, a computer algorithm is able to basically paint in each branch of neuron with a different color. And it can do this not just in a single slice, but in 3D. It can then do this over the complete extent of the data set, such that then if you just go and click on one of these neurons, it'll show you its complete branching pattern. Now, this algorithm is not perfect. There definitely are still mistakes that are made, and it requires still potentially months, years of human effort to go and reconstruct the couple of errors that the algorithm makes, but this takes the amount of time needed to reconstruct a single neuron down from days to something like an hour. So it's really an order of magnitude improvement in how fast we can reconstruct neurons. And with this, we're hoping to get this more complete map of all 20,000 neurons in the fruit fly's ventral nerve cord. And then really, we hope we can dive in and start asking really specific questions about where are all of the reflex arcs? Where are neurons that could be central pattern generators, which is something that has been a big mystery for a long time. And so we're really excited to move towards this more complete reconstruction of the ventral nerve cord using the EM data set that I helped generate. Okay, so I told you the goal of my project is to completely map the adult fruit fly motor circuit. I told you about the efforts using electron microscopy to map the ventral nerve cord. And now I'm going to tell you just briefly about what we found and how we did this of using X-ray imaging to look inside the leg. So the leg is quite a lot larger than the VNC. You might initially ask, well, why can't you just use the same electron microscopy technique and slice up the leg? and image it. And it's because it's a lot bigger. And so imaging time for my VNC data set was about 2 months. If you try to do this for a leg, it would take something like 8 months, which is getting pretty prohibitively long because things can always go wrong during the process. It's also not actually clear that you can even do this kind of technique on a leg because it requires slicing. And the exoskeleton around a fly's leg make it actually really difficult to slice through. And so we needed an alternative approach. And we worked with Alexandre Pacrianu at the European Synchrotron in France to do X-ray imaging of a leg. And I'll tell you about how that works. So a synchrotron is one of these huge particle accelerators. And it turns out that these particle accelerators generate really high intensity electron beams that biologists and physicists then can use for imaging different things. So we take this really high intensity X-ray beam from a synchrotron, focus it down to a point using these special mirrors, and then pass it through a sample. This sample here is a fruit fly's brain, ventral nerve cord, and its front 2 legs. And we can take images then that look like this, which again in 2D don't really show you the 3D structure, but we can then rotate the sample and take pictures, X-ray pictures of the sample at all these different rotational angles to get kind of a 3-dimensional, this now gives us the depth perspective we need. And after processing those images, we can then get an actual 3D imaging volume that reveals what's going on inside the leg. And so we achieved about 100 nanometer resolution with these images over fairly large fields of view, which is about what you need not only to resolve some of the larger structures in the leg, like muscle fibers, the exoskeleton, but also single neurons. So here you see this bundle of motor and sensory neurons traveling through the leg, and you can make out the profiles of many individual neurons so that we can then follow them throughout the leg and see where they connect to. And I won't tell you about all the details, but this enabled us to generate this kind of improved parts list of actually what is in the leg. We found, similar to the EM project, we found some things that people had found before, but we also found a couple of new things that we're excited about. And I'll just tell you about 1 or 2 of those. So motor neurons, they connect to muscle fibers in the leg. Muscle fibers pull on tendons, and then tendons pull on joints. Here the joint is out of the frame, but otherwise the coloring matches. And understanding the connectivity of these is important for really understanding what the ventral nerve cord is controlling. And we were able to see that motor neurons and muscle fibers are organized in these motor units, which is something that people have definitely seen in vertebrates and other animals before. But it means that single muscles are not actually really just single muscles, but they have subdivisions where, for instance, one motor neuron only innervates these 2 lighter colored muscle fibers, whereas 2 other motor neurons innervate these 4 darker colored muscle fibers. And so this means that these 2 different parts of the muscle are under distinct neural control. And we think understanding more about the organization of these is going to be important for understanding, okay, what is the ventral nerve cord actually doing when it's going and stimulating muscles in the leg? One other example is that the X-ray data set shows us what different sensory organs look like. And so in collaboration with John Tuthill's lab, we've been looking at one of the sensory organs in the leg, the femoral cortitonal organ, where there are a bunch of sensory neurons that are placed essentially near the fly's elbow, and then they detect movements of the fly's wrist. And it turns out that these sensory neurons placed near the elbow are connected over to the wrist by these 2 long tendons, which you see here, and they're connected to this funny structure over near the wrist that moves when the fly bends its wrist. And we think this is this is something about how these sensory neurons actually end up being able to detect the movements of the wrist. And so that's a work in progress. Okay, so looking forward, at this point, I've told you that we are starting to get a really good handle on what do all the parts look like of the motor system in the fruit fly. And that's been really exciting. But the next kind of question is, what does each part do? Certainly, previous studies have mapped out what many of these different parts do, but we're still far from having a complete sense of what all the different parts do. And so while in my project, it's been really exciting to map out and see all of these parts, it's been a little bit frustrating not to be able to test out how each part is actually involved in behavior. And that's why for my next job, I'm going to be moving to Pavan Ramdia's lab in Switzerland to study which of the neurons in the VNC are active during which types of movements. Even once you know what the parts do, it then is another step harder to figure out how does the whole system work together. But if we can manage to do this, then hopefully we'll be able to use what we learn to build more agile robots. And looking forward to even larger animals, as I said, the field of neuroscience is really interested in mapping out nervous systems of larger and larger animals. And many of the techniques that I told you about today can be applied equally well to other species. In fact, as a side project I didn't have time to tell you about, I mapped part of the mouse's visual thalamus in collaboration with Lucas Cheadle and Mike Greenberg's lab. Also, I'm the only student or postdoc in my lab studying the fruit fly. Most of the other people in the lab are studying the mouse by applying similar X-ray and electron microscopy techniques. And so it's been really cool to see these similar techniques applied to many different systems to answer many different kinds of questions. So with that, I'd like to thank all of the people who have helped with my projects, members of the Lee Lab, members of John Tuthill's lab in University of Washington, Seattle, who have been really indispensable collaborators in figuring out what questions to ask. Alexandra and Peter at ESRF, the members of Zeta AI and Jan Funke's lab, who have helped with some of the automated reconstructions, and then some other people at Genelia who helped me out. Okay, now for some acknowledgement slides. So overall, I've had a great time in grad school, and a large part of that was thanks to Wei, who managed to foster a really positive and tight-knit environment that made it possible to do exciting science with a really great team of people. So before COVID, our lab had lunch together almost every day, and I have so many good memories of going out for celebration lunches as well. We had a couple of lab retreats up in New Hampshire that were just incredibly enjoyable, and I love that Wei is supportive of these kinds of efforts. We did this in 2 different summers. We had a fun trip to a conference in Berlin, really fun being there with Alexandra, Logan, Aaron, and Wei. Logan and Aaron and I did some sightseeing in Berlin that was really fun. And Logan got this photo of me and Aaron falling asleep on the airplane back in sync. Wei also let me go and visit John Tuthill's lab in Seattle a couple of times on 2 different summers. And that was a really great experience, getting to know everyone in that lab better, as they were really close collaborators on my projects. Finally, once COVID hit, we were still able to stay entertained, for the most part through video calls or making up silly games. For instance, we had this really fun emoji battle bracket that I'm still bitter about how the finals went, but that's all right. It was really fun. We still managed to see each other in person occasionally with outdoor lunches. I also just wanted to quickly shout out some of the musicians who have really helped me stay sane through the past year of working through home: Seven Lions, Oliver Heldens, and Netsky. But returning to the best part of working here is really just the people. Our lab works with a lot of co-op students from Northeastern University who do 6-month positions in our lab, and working with them has just been a treat. Seeing many of them go from really not knowing that much about neuroscience to being able to make really important contributions to the projects going on in the lab is just incredibly fun and a great experience to work with them. And I want to especially shout out Alec, Mingwan, and Leah, who did most of the reconstructions for my EM data set. The co-ops are great. They manage the meme wall. They're constantly posting memes on Slack, and the best ones get posted on a physical wall. And the team of people that I was able to work with to do the X-ray project was also just really fantastic. Getting to know all of them and work on developing a new imaging technology and applying it to neuroscience was just a really fun time. I was lucky to be advised by many, many different faculty members. Of course, Wei, my primary advisor, and then Rachel Wilson, who is the head of basically all of my committees. Both of them took so much time to really help me with my research projects and make sure that I was on track. And I just am so grateful for all of the effort they put towards that. Other collaborators in the department: Lucas and Lauren, other Penn students: Willie, Hannah, Ivan, Asa, Chiyu, Julia, Shi, Rich, and others. And then the admins of the program: Susan Jackson and Karen Harmon. I want to especially shout out Karen Harmon. I was lucky that the Lee Lab is just past Karen's office. And so almost every day on the way into work and out of work, I would pass by and chat with her. And she really was just a support system for me, but also a really just close friend that I was incredibly lucky to be able to work so close to. And so, yeah, just huge shout out to Karen for everything that she did over the years for me. Looking outside of the neuroscience community, the indoor soccer community was really just a fantastic source of exercise and fun for me. Special shout out to Manav for starting the community and for being a good friend, Shahar for keeping things organized, and Kevin for walking me to the emergency room when I dislocated my shoulder. As Wei said, I've also been involved in coaching and running the Harvard Table Tennis Club, where I met a lot of new people and good friends. Special shout out to Austin Duenas and Justin Lin, 2 Harvard Law students who are now good friends. Then my family. So my mom, dad, and sister Sierra have always been there for me to support me, and that's meant the world to me. And I really can't wait to be able to go on vacations together again. Dad, thanks for inspiring me to become a scientist like you. And I really can't wait for the next time that we can go to a table tennis tournament together. To Grace, I met Grace about 7 years ago in a research lab, and we've been together ever since. And every day I still think about how lucky I am to have you in my life, and I'm looking forward to our future together. To the Phelps family, thanks for welcoming me to spend holidays with you. Thanks for all the kindness that you've shown me. And also thanks for letting me use the name Phelps on my papers, even though I'm not legally part of the family just yet. And finally, Prim and Tallulah, the world's best cats. So Grace and I fostered these cats when we were in Chicago, and we loved them so much that we drove them to Boston with us when we started grad school, and that was the best choice ever. They are just pure bundles of joy, and they fully deserve this slot as my last and most important acknowledgement slide. And so with that, I'll stop, and I would be happy to take your questions.
Thank you, Jasper. So people are now able to unmute themselves, and we do have a couple questions already in the chat. The first one was from Davi Bach. Davi, would you like to ask your question verbally?
Yep. Hi.
Yep.
Hi. Nice work, and thanks for the talk. I was curious if you could compare what you're seeing in adult VNC to that of larva, where a great deal of work has been done. And I was interested in the distinction you drew between a limbed versus limbless body plan. And I'm wondering how you see these limbed cell types integrating with the perhaps a limbless structure established in the larva.
Yeah, absolutely. Let me go back to the slide. Yeah, it's been very cool to think about kind of comparisons between the larva and the adult. For people who don't know, so the fruit flies go through this larval stage where they don't have any limbs, they just eat a bunch of food, and then they go through metamorphosis to grow limbs. And the nervous system gets about 10 times as big, but many of the neurons present in the larval nervous system are then present in the adult nervous system as well. So one of the differences that we drew is that direct connections from sensory neurons to motor neurons are pretty infrequent in the larva. Whereas in the adult fly, I showed you that we have at least this one very special cell type that makes extremely strong direct connections to motor neurons. And so that this really fast monosynaptic reflex arc probably underlies some kind of a fast movement. And fast movements may not be super necessary when your body is just kind of a tube that doesn't really have the ability to do fast movements. And so it's possible that in the adult, where its body physically is capable of doing faster movements, there might need to be neural pathways that are faster in order to actually make use of those much faster body movements. And monosynaptic sensory to motor connectivity might be one way to have these kind of faster pathways. There's also been a lot of work looking at, the moonwalker descending neuron, this neuron that makes both the larva and the adult fly walk backwards when you activate it. That neuron is present both in the larva and the adult, but somehow in the larva, it has to cause backward crawling movements, and then the adult, it causes backwards walking movements. And so there's a lot of interest in these kinds of things. Yeah, so thanks for the question.
Sorry, I was muted. We have another question from Jihao Zhang. Would you like to ask it?
Yeah, congratulations. Nice talk.
Thank you.
I'm just curious about the interneurons. There's a catalog of mononeurons, and we're making headway into the sensory neurons. Looking at, just roughly by the math, it looks like probably more than half on interneurons. How much do we know about the interneurons from this data set?
So I haven't looked at the interneurons very much yet, but that's absolutely where we're going next. We started from the periphery, sensory and motor neurons, and are kind of working our way inward. There's a lot of literature about what different interneuron types are in the VNC. We know that the developmental lineage of these different interneurons determines a lot about their morphology and their connectivity. And we've already started to correspond the interneurons that we've reconstructed with those previously published, known lineages of neurons, many of which have relatively known functions. But we know almost nothing about the connectivity patterns between the interneurons. We know a little bit, but that's kind of one of the avenues that we're really interested in going down, because the interneurons come from these very distinct lineages, and we know a little bit about their function. Going and mapping connectivity patterns between interneuron lineages is something that we're definitely interested in working on.
We have a question from Farzan Salman. Farzan, would you like to ask a question, or I can read it? Oh, no mic. So Farzan's question is, well, he says, great presentation. I was wondering what role wings / flight play in motor connectivity.
Yeah, absolutely. So fruit flies, obviously they also fly. And in my talk, I focused a lot on limb control, both because I haven't yet heard of a human who has lost their ability to fly. And also we have robots like helicopters, things that are propeller based, that fly really, really well. And so my interests are definitely more in the limb circuit, but certainly other members of our reconstruction community are using this VNC data set to map out circuits that control flight, and are answering a lot of questions like that as well. In terms of like direct interactions between leg control circuits and wing control circuits, Gwyneth Card's work has looked at things like takeoff behavior when flies jump and then start flapping their wings. And so certainly there are neurons that need to coordinate movements, whole body movements that involve both the legs and the wings. But that's something we haven't really directly looked at yet. But hopefully this data set will be useful to people who are interested in those kinds of questions. I see Rick has his hand up.
Yes, Rick.
Hey, Jasper, just gorgeous work. I really enjoyed the talk. I think of connectomes as kind of the ultimate hypothesis generating engine. And I guess I'm wondering if you could speculate on some of the more interesting hypotheses that your data set might suggest. I guess I was fascinated by those 12 neurons, sensory neurons that are bilateral and make immediate synapses onto motor neurons. You suggested that they were weight sensors, and I'm just wondering, do you think that's like an anti-gravity system, or what other hypotheses have you generated?
Yeah, absolutely. We've talked a lot about like what the heck could these neurons be doing. So what we know right now is that they probably detect change in weight, and they probably cause flexion of really forceful flexion. And we think that they make that happen on multiple limbs at the same time. And so we're imagining something like this. So maybe this is a recoil response, or maybe if the flies are about to be like blown away by a gust of wind, they grab the ground. We don't know exactly what it is, but we have a couple hypotheses about, maybe it's a recoil, maybe it's a grab the ground kind of response that we would love to test. In terms of more generally, I totally agree that connectomes basically show you all the pathways. They don't show you exactly what they do, but properties of those pathways, where the connections are, what cell types are involved, can give ideas about, where could this information be flowing from sensory to motor. Also down from the brain. I didn't talk too much about this, but there are also this big bundle of connections coming down from the brain that people can look at and use that to generate hypotheses about how different descending signals coordinate movements of the body.
Brittany Scheid, you had a question? Would you like
Brittany,
to
long time no see. Can't hear you, muted.
I didn't do the wrong one.
Thanks
for inviting us, and this was really, really beautiful. So my question, I've actually always been wondering this when I hear about connectomes, is how much variability is there between individual flies as far as our connectomes go? It seems like the structure was pretty well conserved because you were able to see, the neurons that were already found from prior work. But I'm wondering if the variability is small enough that, maybe if there's a mutation, you can actually see some differences between a normal versus abnormal connectome structure in the future. But I've always been curious about the variability question.
Absolutely, yeah. And there was previous work by Wei and Rachel Wilson looking at when there are kind of asymmetries in the nervous system. For instance, we see that there's a difference of 1 on the left side and right side of the nervous system. And how does that change the connectivity patterns that you see of those neurons? And they did some previous work using electron microscopy to map out two different sides of the brain where there was a clear difference, like one of the neurons just didn't develop or something, and they looked at how the connectivity adapted to that. And in that system, it was clear that the existing neurons kind of took over a lot of the connections that would have been grabbed by the other neuron if it had developed. But yeah, the question of variability at the connectivity level is definitely something that neuroscience in general is really interested in. There have been definitely some comparisons between different data sets. There was recently some work in the olfactory system from Greg Jeffress's lab. So connectomics has really been at this kind of N of 1 state for a long time, where we can get one data set, but getting a second one is tough. And you need that in order to answer these kinds of questions of variability. And it's exciting that the imaging technology is now kind of reaching a point where those kinds of things are possible. And so I bet you anything that in the next 10 or so years, we're going to start having really, really good answers to those kinds of questions. There already are some senses that like the strongest connections are pretty reliably present in multiple animals, whereas the connections that maybe only have a couple of synapses, those may not be there in every animal. But yeah, almost certainly more results about that in the coming decade.
Awesome, thanks.
Yeah, thanks, Brittany.
Bruce Phelps.
Yes, good morning, Jasper. Excellent presentation.
Thank you.
Enjoyed it a lot. This is more of a high-level question. I would think the neuronal circuitry would be incredibly important for the survival of an organism, sort of the efficiency and optimality of that circuitry. So I was wondering if you could maybe just, give me your views on sort of the evolutionary pattern of this circuitry. And the reason I asked this question is I was thinking about the robots that you showed at the beginning, and I'm wondering if given your research, might you think differently about building robots rather than relying on what we observe in these organisms?
It's certainly true that efforts to just build robots without really caring about animal nervous systems may end up, just continue to work really well. And so, there are these definitely two different approaches to building intelligent systems. One, you just kind of do it based on the principles that you think are important. And then the other is look at what nature has built and try to build it based on similar principles. And it's not totally clear which of those is really going to get us to kind of landmark things like, general artificial intelligence or really skilled robotics first. As far as evolution of the nervous system, absolutely. A lot of these neural circuits evolved because evolution has put pressure on them too. One of the things we're really interested in is with this new pathway from the BCS neurons to the motor neurons, what was the evolutionary pressure to develop that? As far as we know, larger insects like stick insects don't really have this type of neuron. They move a little bit slower, so maybe that's it. But yeah, thinking about what was the evolutionary pressure that caused a particular bit of a circuit to develop is a really interesting question. It always comes back to behavior, right? Neurons only exist in order to generate some behavior. And so kind of a unit, the thing that evolution acts on is, did your behavior get better or worse? And there's a lot of people who think about how neural circuits, how the evolution of neural circuits leads to the evolution of behavior. And so, yeah, it's an interesting field. Yeah.
Are there any other questions from the audience? If not, we'll move to the closed portion of the defense. Though, if people happen to be in the Boston, Cambridge area, at 1:30, we'll be meeting at Dana Park in Cambridge for a little gathering to congratulate Jasper on his defense. So with that, we will move to the closed session, unless there are any other last questions.
Thanks everyone for coming. Really great to see you all. Please feel free to send me an email if you have any questions or want to chat. Really fantastic to see you all here. Thanks for being here.